What is the double helical structure of DNA and why was its discovery important? Well, I think that there's barely a week goes by these days when we don't hear something about DNA in the news. A few years ago it was being used to identify the body of Richard III. Just recently I was reading a story where it was being used to verify the authenticity of entries in a vegetable show. More seriously, it's been used for DNA fingerprinting in crime solving, and it's given rise to new medicines like recombinant insulin for the treatment of diabetes. And at the centre of all this technology is our understanding of the shape of the DNA molecule, and this is the double helical shape. So what that means is we have these two chains of sugar and phosphorus twisting round into this helix, and going up the centre we have these four different compounds shown in four different colours here called the bases. And it's the bases that carry the genetic information that's passed on from generation to generation. So it's the base sequence that dictates whether we're going to have blue eyes or brown eyes, black hair, fair hair, or sometimes no hair as well. The structure, the double helical structure, was proposed by the two Cambridge scientists, James Watson, Francis Crick, in 1953. And the reason that was such a milestone is this double helical structure explains how the molecule can copy itself, and that's the one thing it has to do if it's going to be the genetic molecule, if it's going to pass information on from generation to generation. It has to be able to make copies of itself, and that all comes from this twisting double helix. So who was Astbury? Well, Astbury was a physicist, and he'd come to Leeds in 1928 to use X-ray scattering techniques to study the molecular structure of wool fibres, because Leeds was the centre of the textile industry, well, at least the wool industry. So Astbury began with studies of wool. From that, he established himself as an international authority using X-rays to study the giant molecular fibres from which living things are made. And he established himself as a pioneer of the emerging new science of molecular biology. That is, the idea of understanding living systems in terms of the shape of these giant molecules from which they're made. And through that work, he began in 1938 with his research assistant Florence Bell to make the very first attempt to solve the structure of DNA using X-rays. And so Astbury's work on DNA culminated in 1951 when Beaton took this image that was almost identical to photo 51. But the curious thing is, whereas Rosalind Franklin and Raymond Gosling's photo 51 made Watson's mouth fall open and his heart race, Astbury did nothing with Beaton's photograph. In fact, it was the end of his work on DNA, and he never did anything else on DNA after that. And I think the reason that Astbury gets forgotten is because if you focus solely on him and DNA, you actually miss out on what his real scientific contribution was. And I think that was this idea of understanding living systems in terms of these giant chain molecules, and also the idea that we could perhaps deliberately alter these chain molecules for our own end. And I think a great example of that was in his famous monkey nut overcoat that was made out of proteins taken from monkey nuts whose shape had been deliberately altered.